Welcome. In this video, we are going to talk about peptide and polypeptide. Now, peptide is a compound that consists of two or more amino acids. Okay, a peptide is a compound consisting of two or more amino acids. Now, when two amino acids are linked by a peptide bond, then that compound is known as dipeptide. Okay, what is dipeptide? Dipeptide is a compound which consists of two amino acids linked by peptide bonds. Now, if three amino acids are joined by two peptide bonds, then that compound will be known as tripeptide. Similarly, the amino acids can be further linked to form tetrapeptides and pentapeptides and so on. Now, if your peptide chain consists of more than 12 amino acids but less than 20 amino acids, then that peptide chains are referred to as oligopeptides. So, oligopeptide is a compound which contains more than 12 amino acids and less than 20 amino acids. Okay, and the thing is that some peptides are cyclic as well. Okay, now for example, there are these two cyclic decapeptides which are produced by a bacteria known as Bacillus brevis. Okay, now these two cyclic dipeptides are gramicidin S and tyrosidin A, and both of these decapeptides that are produced by this Bacillus brevis are antibiotics, and both of them contains D amino acids as well as L amino acids. And these two peptides also contains ornithine in them, which don't usually occur in a protein. Okay, so these are the two examples of the decapeptides which are produced by this bacteria Bacillus brevis. Okay, now so this small peptides which we are talking about have many roles, many important roles in an organism. Okay, consider this oxytocin and vasopressin. Okay, these two are the small peptides only, and they are important hormones. And this glutathione is also a small peptide which regulate the oxidation reduction reactions. Then you have this enkephalins. Okay, it is also a small peptide which is a naturally occurring painkiller. Now this aspartame, okay, it is a commercially synthesized dipeptide. Okay, it is known as L-aspartiphenylalanyl methyl esterase, and this thing is used as an artificial sweetener. Okay, so it is also a dipeptide, peptide. Okay, so peptides are important. That's what the point is. Now, when many amino acid residues are joined, then there is formation of the compound which you call as polypeptide. Now, the amino acid that have been incorporated into a peptide or a polypeptide, then we call that amino acid as amino acid residue. Okay, so when amino acid is not joined with other amino acid, okay, it exists as single, then we call it as amino acid. But as soon as it gets joined, it gets incorporated, and there is formation of peptide and polypeptide from it, then that amino acid in a peptide we refer it to as amino acid residue. Now, by convention, in a polypeptide, the left end represents the first amino acid and the right end represents the last amino acid. Okay, so whenever you are drawing a diagram, okay, now by convention, it is fixed that the left end of that polypeptide diagram is going to represent your first amino acid, and the right end of that polypeptide is going to represent the last amino acid. Now, the last amino acid of this polypeptide is also termed as N-terminal amino acid residue. Okay, so this is how the structure of a polypeptide chain looks like. Now, as you can see, this portion, then this portion, then this portion, these are the individual amino acid residues. Okay, now this end is your N-terminal, which is representing your first amino acid, and this end, which is your C-terminal, is representing your last amino acid. So like amino acid, the first amino acid is having free alpha amino group, and the last amino acid is having a free alpha carboxyl group. And that is what is represented in this diagram. Okay, so at C terminal you have carboxyl group, at N terminal you have amino group, and these are the individual amino acid residue structures. Now the peptide bond between the in proteins are formed between the alpha amino group and alpha carboxyl group. Okay, so between alpha amino group and alpha carboxyl group there is formation of the peptide bonds, and that is how one amino acid is linked to the other amino acid, and at last there is formation of proteins. Now several peptides are there, they occur naturally, in which there is a peptide linkage between a carboxyl group or amino group which is attached to a carbon atom that is not alpha carbon. Okay, in the ribosomically incorporated amino acid you saw that the carboxyl group is attached to alpha carbon and the amino group is also attached to alpha carbon, but there are many naturally occurring polypeptides, peptides which don't have this carboxyl group attached to alpha carbon, okay, or amino group attached to this alpha carbon. It may be attached to some other carbon, and even if it is attached to some other carbon, the peptide linkage exists between that carboxyl group or amino group. Okay, so consider this dipeptide known as gamma glutamethylαlenine. Okay, now in this case of dipeptide, the peptide bond is between the gamma carboxyl group of glutamic acid and the amino group of alanine. Okay, so now this carboxyl group which is involved in the peptide bond formation is not attached to the alpha carbon, it is attached to gamma carbon, and that is the reason why it is known as gamma carboxyl group. And this is the example that it is not necessary. Okay, there are some naturally occurring peptides as well, in which the peptide bond can exist between the carboxyl group or amino group that is not attached to the alpha carbon. It might be attached to any other carbon, and the example of that thing is your gamma glutamethylαlenine. So that's all for this video. Thanks for watching.